the research of outstanding graduate students in the biomedical sciences. The College of Letters and Science is very grateful to Phil and his family for this continuing support for students, as well as the Kopriva seminar series that enriches the intellectual life of our campus. His legacy lives on through these gifts. Today, we get to honor both aspects of this gift in that the speaker was awarded a Kopriva Graduate Fellowship and is today presenting a talk as part of the Kopriva Science Seminar Series. To introduce the speaker is Beth Burrows, head of the Department of Mathematical Sciences. Beth, if you would.
Thank you, Ives. Hi, everybody. I am very proud to introduce you to Catherine Potts, who is today's speaker. Catherine is a PhD student in the Department of Mathematical Sciences. Her research is in the area of machine learning and geometric data analysis, and she's focused on scientific imaging applications. She is nearing the end of her PhD program and will defend her dissertation this fall. She's been part of the Montana Peaks Fellowship Program here in the department, and through that, she's worked in a neuroengineering lab, and that work she has done is the focus of her talk today. She has been very active in our department. She's an organizer of the Rocky Mountain Data Science Club and has been for over 2 years. She has had an internship at Los Alamos National Labs working in applied machine learning. And since 2020, she has worked as a student intern for Quantum Computing Incorporated. So we are very proud of Catherine and just really interested to hear what she has to talk about today. So with no further ado, Catherine Potts.
Thank you. Thank you so much for those wonderful introductions. I'm very excited to talk about what I've been working on for the last few years today. The title of my talk is Archetypal Analysis Applied to Neuronal Cellular Signaling. And like Beth mentioned, this is work that was part of my Montana Peaks Fellowship, and also in collaboration with the Kunze Neuroengineering Lab here on campus. So let's go ahead and get started. First thing I want to talk about is neuronal cellular signaling and what those words mean. So here's an example of a neuron, and a neuron is a cell that is in your brain, so a brain cell. And we have these cell bodies here just as an illustration. And when they want to communicate with each other, what they'll do is they'll send an electrical pulse through these axons, and then one neuron will send the pulse and the other one will receive it, and that sends some information between the two cells. So this pulse is sent as an action potential. However, the action potentials have a very short kind of wavelength here, so they aren't always easy to pick up, and they're not easy to pick up the way that I would like to pick them up, since my research is based on imaging problems. So difficult things to image. However, there's a secondary response when an action potential is sent by the cell, and that secondary response is in this release of a calcium ion here. So this calcium ion has a longer wavelength, so we can pick it up a little bit easier in an image. And how we do that in the lab is we introduce a calcium dye to the neurons that we grow, and then we can image that calcium ion using that dye over time. So when we do that, we get a series of images. Each image looks something like one of these images here, where we can see the cell body. We can even see a little bit of some axons between them. And through time, what happens is when an action potential is released by one of these cells, the calcium expression increases, and what we see is these green cell bodies get brighter, and then they start to decay after a very short period of time. And when that happens, we say that the cell has fired, so has sent that action potential to another cell. Traditional means of analyzing these sorts of calcium videos, I should say, so we record over a period of time. So we're getting really a video of each of these cells as they fire. And traditional methods look at individual cells and they assess the firing behavior of individual regions of the video itself. However, there's a lot of interest in what is happening kind of collectively. So which cells, for example, are firing at the same time, or which cells are directly communicating with each other. And that can be a little bit difficult to do if you're looking at just an individual region itself. So our goal is to, instead of look at individual cells and on the back end assess when they fired and how that relates to the other cells, our goal is to do that kind of at the same time. And so we can kind of pick out collections of cells that fire in unison. So just to sum up, that's what our analysis goal is. We want to pick out these neuronal cliques. So these are the neurons that are firing in unison with each other, which may indicate some sort of network connection. We also want to say when those are firing. And so what we have been working on is, again, taking these calcium videos and looking at the cells, trying to pick out these cell cliques and saying at what time in the video each cell clique is active. So that's our goal. And we're going to use some unsupervised machine learning to accomplish that goal. So just a little bit about the data we're applying this method to. First off, the video frames are 2200 by 2700 pixels. We have about 7,000 frames for each video, and they can take up a lot of space in memory on certain computing platforms. For example, we can think about every single one of these video frames as a data point, which means that every single piece of data that we have here lives in our 6 million approximately. So very, very large, high-dimensional data. And like I mentioned, the data can be too large to store in core. It may be too large for certain machine learning methods, such as matrix factorization. And another challenge that we're facing here is we really want to capture the biological significance of these cellular videos, because again, we want to pick out the behavior, we want to pick out when cells are firing in unison with each other. So those are kind of the things that we have to tackle with our analysis. And so, let me talk about how we went about those challenges. So what we, let me go back. So what we chose to use for our machine learning model is archetypal analysis. It's not super common, but I'm going to explain it, so don't worry. And first off, let's kind of start with a little bit of an example. So let's say every single one of these kind of pink data points here, let's pretend that they represent some image in a very high dimensional space. So this is kind of our data cloud, and we're thinking of each one of these points as an image. And then what we can do to kind of encompass this data cloud and actually model this data cloud is calculate its convex hull. So for example here, the convex hull is going to be the polygon enclosed by those orange lines, and it's defined by these orange data points here. Those are called extreme points of the convex hull. And what's cool about these extreme points is that every single one of these pink dots can be represented as a convex combination of these orange points. So instead of needing all of this information to model our data, we can just use our extreme points and then some information about how to combine those extreme points together, and we've recovered all of our data. So that's kind of exciting. And let's talk about how archetypal analysis uses this. So first off, there's a little bit of math on this slide. I promise there won't be tons and tons of math going forward, but a little bit at the start. So let's say we have a given data set in our M. Remember, we're thinking about M as being 6 million, and we have N pieces of data. We're thinking about that being like 7,000. So our archetypes are going to be a weighted combination of each of these original data points. So archetypes are also going to have 6 million pieces of information contained in them. And we're going to use a very special sort of combination. We are requiring the combination to be a convex combination. That means that each of these betas must be positive, and when we sum them over k, they have to equal 1. That gives us a lot of really nice properties. Basically, what that says is that our archetype zj is going to look very similar to one of our data points. So that's nice because we can interpret our archetypes in the context of our data. So that's what our archetypes are. We can reconstruct our data points using the archetypes with just another weighted sum, where again, we have the same restrictions on our weights. They have to be non-negative, and they have to sum over j to equal 1. So we have another convex combination here. And what we want is to choose the alphas and the betas so that this xi hat matches the xi data point as close as we can. A couple other things I wanted to point out. Notice that, remember, we have n archetypes, and we were thinking about that n as being 7,000, but we only have n data points, and n is 7,000. We only have p archetypes. And so ideally, we'd like to choose p to be much less than n. So we have a smaller kind of representation of our data in the model. Okay, so that doesn't quite help us choose the alphas and the betas. To find the alphas and the betas, we solve this constrained minimization problem, where the A matrix holds all of our alphas, and the B matrix holds all of our betas. The X matrix is each of our data points arranged in as columns of a matrix, and then this XBA here is going to be our reconstructed data points. So we want to minimize the difference between our reconstruction and the original data, and we minimize over A and B. And we also still require the convexity constraints that we had before. So looking at this, this is a form of a constrained optimization problem, like I said. It's also a matrix factorization machine learning method where we have XB will be our archetypes, and the A matrix will say our reconstruction weights. So those hold all the alphas. Okay.
Can I ask a quick question?
Yes.
Is P fixed, or are you going to actually optimize over P?
So model by model, P is fixed, but you can certainly run your model multiple times to optimize P.
Got it.
Okay. And let's actually look at what happens with P. So here I've applied archetypal analysis to that earlier example of the data cloud that we were thinking of images. And in this first example, I've chosen a 3 archetype model. So here P is equal to 3. And what we see is our archetypes will form a convex hull that looks like a triangle. Whereas if we choose P to be larger, we'll completely encompass the entire convex hull of the original data set. So we cover all of the orange lines basically here. So the more archetypes we choose, or we choose to use, the higher we expect to get a complete picture of our data. But sometimes that's not necessary. Sometimes it's okay to kind of just look at larger trends, and so use a lower P. I should also comment that each of the data points inside the triangle will contribute no error to the model itself. However, all of the data points outside of the triangle are being projected inside the triangle in their representation, in their reconstruction. So each one of those is going to contribute some model error. Okay, I promised that for those of us who know what principal component analysis is, I promised that I would compare archetypal analysis to principal component analysis. Principal component analysis, one way to express that machine learning method is by this unconstrained minimization problem as a matrix factorization method with no constraints. And so this is very similar to archetypal analysis, except that archetypal analysis has those convexity constraints, which makes it a little bit more complicated. Here, the Z matrix has the principal components in it as columns. And in certain cases, we can interpret the principal components as the eigenvectors of the data covariance matrix. And really what that means physically is that the principal components are going to point in the direction of largest spread of our data. Also, since we can interpret them as eigenvectors, they're orthogonal to each other, which means we need less of them to cover a larger reconstruction space. And so let me show you what I mean by that. Here's two examples of principal component analysis and archetypal analysis applied to the same data set, just put side by side. So every blue circle is a data point. The orange dots are those data points being projected into the data model, whether that's principal component on this side or archetypal analysis on this side. Here, on the principal component side, we have two principal components used in this model, and we can see their direction applied here. So the vectors are moving in those two orthogonal directions, and the model that we're representing is all of these points kind of in the plane that span by those two vectors. And then on the other hand, archetypal analysis, on this archetypal analysis side, we have a 3-archetype model. The archetypes are going to be the corners of this pink triangle, and all of our data is getting projected into that triangle. So on both sides, the black lines represent the model error for each individual data point. So if you think about adding up all of those model errors, that's going to be the total model, the total error for each type of model. Principal component analysis, we're just projecting right down, straight down into that plane. Whereas archetypal analysis, we have to kind of like project at a diagonal in some areas. So we actually kind of expect here that we would get more error with archetypal analysis than over here with principal component analysis, even though we use more computational elements in archetypal analysis. So you hear that and you might think, whoa, Catherine, like why would we do this? Okay, you're telling me that we need more components and that the error is going to be larger. That doesn't sound like a great machine learning model. Well, we're doing it because we're using archetypal analysis because it has a really awesome biological interpretation, especially for imaging problems. As I mentioned before, each of the archetypes looks like an original data point. So if we have this data set here, this set of faces, we can look at principal components compared to archetypes. The principal components are these images here. And they look like you can kind of see a shadow of a face, but I wouldn't expect to see someone looking like this just walking down the street. Whereas the archetypes look exactly, well, some of the archetypes look exactly like a human face. And another important thing is that if you notice this guy right here, this archetype down here is a person that's wearing sunglasses and a mustache. And we actually have a person wearing sunglasses and a mustache represented in our data. So this archetype is picking up on that trend in the data. Now, I would argue also that this trend may be kind of an extreme trend in our data, but that's not necessarily bad for our purposes, because we want to think about firing events in a large imaging set, and firing events perhaps are not the most common trend. So we do want to pick out those sorts of things that we may think of as kind of a extreme trend in our data. Okay, so this is why we're using archetypal analysis. We get a lot of biological interpretation. Now, let's talk about sketch-based archetypal analysis, which is one of the main pieces of my dissertation. So again, let's remember what we were working with here. We said we needed a model that captures biological significance, so we've chosen archetypal analysis because of the biological interpretation that we get. But we still have data that might be too large to store in core and maybe too large for matrix factorization. And that's a big one because archetypal analysis is a form of matrix factorization. So what we have is super high dimensional data. Remember, we're thinking about each image as being in R6 million, and we have 7,000 frames. Now, one of the common assumptions for imaging data is that the surface that generates the images in your imaging data set is much lower dimensional than the embedding dimension. So we don't necessarily need 6 million pixels to represent each of these images. So the idea here, or one idea here, would be to take that data point in R6 million and project it into a lower dimension. And that would help with both of these points, the data being too large to store in core and too large for matrix factorization. So we have to be careful. We have to be careful because we want to maintain the geometric interpretation of our model. We want to remember that our archetypes are modeling these extreme points of the convex hull. And if we project the data, we don't want to lose that interpretation. So we have to be very careful in how we project, because things like this could happen. So for example, if we take this triangle and we project it to its first component, we get a line, which is fine. However, this triangle has 3 extreme points to its convex hull, and this line only has 2 extreme points. So we actually lose an extreme point under this projection, and which is exactly what we don't want to have happen when we're projecting our data into a lower dimension. So we have to be very careful, like I said. Thankfully, we proved a theorem that says that for a finite set of data points, we're thinking about them being in R6 million, there exists a projection map that takes points in R6 million into a lower dimension RD that will preserve all of the extreme points of the data set. So that is very exciting because that means we can use this projection map and not lose the interpretation of how our archetypes relate to the convex hull of our data cloud. So we can apply the projection to an individual data point like this, and I'll call this S1. We'll come back to this notation a little bit later. So there's implications of this theorem. First, that the extreme points of the original data are going to be mapped to extreme points of the projected data. And also, if we can preserve these extreme points, we can have hope of preserving archetypes under projection. Okay, so I'm not going to go into all the steps of proving this, but I do want to talk about 2 very big components of this proof. One is this Johnson-Lindenstrass lemma. And basically what this lemma does is it proves that there exists a map that is going to control the pairwise distances of our data points after we project them. So let me go through that very briefly. F here is our projection map. And what the lemma says is that for every set of n points in our M, remember n is 6 million, there exists some map F that maps 6 million to some RD, so that for each point, or for every two points in the original set, the projection of those 2 points is bounded below by a factor multiplied by the original distance between those 2 points before projection, and above by that same distance, again multiplied by another small factor. So this is very exciting because that means we can look at the points pair by pair and know that their distances are preserved somewhat, even if we project. Also, we have a little bit of a description of what we can project, of how far we can project onto. So the dimension that we project onto has to be order epsilon to the -2 log n. Remember, n is the number of data points that we have. Epsilon is a value that we choose, a small value that we choose. So this gives us kind of a range of what reasonable projection dimensions would be. So that's great. Another very
question.
important,
Quick question. Sorry.
Yes.
Are these maps constructible, right? So the one,
Yeah.
the Johnson and Strauss, and the one which you said there is a map. This is kind of something. Okay.
Absolutely. Yes. They are constructible, and they're actually not too difficult to construct. But I'm going to save that for my defense. Okay. Okay. Another piece that went into proving this theorem is a way to kind of measure how extreme an extreme point is of a data set. So we defined this delta extreme point by saying a point P is a delta extreme point if there exists another point Y such that these two conditions hold. So first, the distance between Y and P is equal to 1. That's not too hard. And then here we want to look at the vector between Y and P, the vector between X and P for every X in the data set that isn't P. And we want the inner product of those two vectors to be less than negative delta, less than or equal to negative delta. So this may seem a little bit mysterious, but it really prescribes two things. So for, let's say this point is our delta extreme point P here. Here is one point X in the set left over. Here is our point Y that we're interested in comparing it to, and these are the two vectors. What this condition tells us, first off, is that this theta, the angle between these two vectors, has to be greater than 90 degrees. And it also tells us that the distance between P and any of these other blue dots has to be no larger than delta itself. So we get this kind of separating band between our extreme point and all of the other data points. And so we can see that this point is pretty far away from the rest of the data set. So our delta is relatively large. However, if we looked at a different data point on the edge of our data set, for example, this one right here, our delta is quite small here. So we have a way of saying this is a very extreme point or this is not a very extreme point using this definition. And so that idea combined with, oops, I lost my, that idea combined with the JL lemma is what allowed us to prove this theorem. Again, I will go into much greater detail in the fall. So that's kind of exciting. We have some theory behind actually kind of going through and hopefully preserving these important points to our model. So let's talk about how we actually use sketch-based archetypal analysis and what that procedure is. So given a projection map and our data set, first we calculate a data sketch. So we take our projection and we multiply it by all of our data set. We could also do this online if we needed to, if the data set is too large, like the example of the calcium imaging videos. Once we've calculated the data sketch, we can solve the archetypal analysis problem by finding this A tilde and B tilde on the sketch data. Of course, this A tilde and the B tilde are still subject to those convexity constraints as the earlier model. Then, once we have the B tilde and the A tilde, we can go through and calculate the archetypes. Again, we can do this online if need be, and use the reconstruction weights as just our A tilde. And then the resulting data reconstruction is going to look like this. So we have our original data set multiplied by our B tilde. So here we have our sketch-based archetypes multiplied by the sketch reconstruction weight. And then the total model error is going to be determined here just by comparing that reconstruction to the original data set itself. Okay, so yeah, let's look at an example of applying this sketch-based archetypal analysis. So remember a few slides ago, I had all of those different images, those faces images, that big data set. Let's say we constructed a projection map and we want to apply sketch-based reconstruction to every single one of those images. So we want to apply the model to those images. So I'm showing one example of one image here. It's 16 by 16 pixels. We can apply a sketch projection. And so we have a sketched image here. It's now 8 by 8 pixels. So we have reduced the size of the data. And then taking the full collection of all the sketched images, we apply archetypal analysis. And we take something that doesn't look like a face and we build a reconstruction here that does look like a face. So that's pretty exciting, but you should watch out, right? Because you may be saying to yourself, well, Catherine, yeah, that looks like a face, but it doesn't look like the face that you started with. And I would say, yeah, okay, yeah, that's fair. That's fair. It doesn't look very much like that face. However, if we compared this reconstruction using the sketch-based archetypal analysis to just a standard archetypal analysis, they look very similar to each other. So there may be some theory there. And in fact, there is some theory there. And we proved this other theorem that says that if you have a sketch projection map that satisfies the Johnson-Lindenstrass lemma on these sets, then your sketch reconstruction model error here is going to be bounded below by the model error without any sketching, and bounded above by a factor times that same model error without sketching. So we're kind of going to be restricted. Our reconstructions are going to be restricted with the sketch-based model to be very near this the model error from the archetypes themselves. So that's why we can see that this reconstruction is very similar to the reconstruction without sketching. Okay. Now let's bring it back to our application, and let's talk about how we used these things and applied them to the calcium videos, the neuronal cellular signaling. So again, remember what our goals were. Our goals were to apply some machine learning, archetypal analysis, so that we're picking out these cliques of cells that are firing at the same time. So archetypal analysis, this is one of the benefits of archetypal analysis, is that we're picking out extreme trends of our data. So we would expect that our archetypes themselves would give us those sorts of clusters. And in fact, we've seen that to be the case in our application. And these are the types of components that we get out of the archetypal analysis model. So here given a video, we have 3 archetypes displayed here. So this is what directly comes out of the model. Here, we've switched the background coloring so you can see the cell bodies a little bit more clearly. So these are the archetypes that we get out. And then these down here, this image depicts the reconstruction weights that we get out of the model, where something that's very dark blue means that this archetype A3 over here is not being used in the video reconstruction. However, over here, this yellow color, A1, means that this archetype is being used, 100% of it is being used in the video reconstruction at that point. So you can see where each archetype is turning on. For example, A3 is turning on around this part of the video. A2 is turning on around this part of the video. And we got some interaction between A2 and A3 that could be firing in between these 2 cell clusters here. Using these reconstruction weights, we also developed this mixture evolution index. And this mixture evolution index basically looks at each column of our reconstruction weights, which tells us how much each archetype is used in one video frame. And we calculated basically a forward difference. So that means that if our mixture evolution index is up here at 1, we've completely changed all of the archetype mixtures. So a completely new set of archetypes is representing the video at that point in time. But if it's down there at 0, that means that it's the same number, the same archetypes with the same concentrations that are being used to display the video on the next frame. So for a firing event, what we expect to see is some cells turning on and then kind of remaining for a short amount of time. So that would indicate for this mixture evolution index that we would see a change, so something very close to 1, and then something very close to 0 as those cells kind of stay at their same brightness for a certain amount of time. So we would expect to see some sort of behavior like this indicating a firing event. So let's talk about, so these are the things that we use to apply archetypal analysis to the biological application. Let's talk about different ways that we validated the model. One is by applying the results to different petri dishes, so different cell cultures growing over different days, different numbers of days. So for example, 7 days here, we saw a lot of things that look like firing behavior. We see very distinct cell clusters, and we can talk about when those cell clusters are active. So for example, archetype 6 is active around, for example, 600 seconds in the video. And also around 600 seconds in the video, we're switching between archetype 6 and archetypes 2, 3, and 1. Archetype 1 looks like a background archetype, and 2 and 3 are kind of competing so we're showing different sorts of clusters than 6, although 2 and 6 look pretty similar. But so you can kind of see what sort of clusters we expect are turning on and which clusters are turning off. On this day, or this cell culture, we don't see a lot of firing behavior, and we can kind of tell that in a couple of different ways. First off, we don't see the strict up and down motion in our mixture evolution index. We also don't see kind of the switching on and off visually in our reconstruction weights here. And also all of the cell clusters look very similar to each other when we look at the archetypes themselves, with the addition of this kind of background archetype as well. Another way that we validated the model was we looked at what happens when we grow a cell culture and image that same cell culture over different time periods. So for example, instead of different days, we did different time periods. So we did 300 seconds, then we waited 300 seconds, and then we imaged again. And so we can see some firing behavior when we start imaging this one, whereas we don't see any firing behavior after, after our second set of imaging. And this is another really important way that we validated the model. We compared it to spike train analysis, which is a state-of-the-art method to assess these calcium images. And so Connor Beck in the lab helped me do this analysis here, where we take a calcium video and we directly compare it to the processing that would be done with spike train analysis. So that sort of processing involves calculating these regions of interest for the cells, and then calculating a forward difference indicating a calcium influx here. And when that forward difference is steep enough, we say that there's been a spike detected in that region. And we can collect those spikes in what's called a spike raster plot here. So based off of the region of interest and then the time here. So to compare that to archetypal analysis, we chose an archetype that closely overlaps with our region of interest here. So we have a good overlap between these two. We looked at the reconstruction weights for this archetype, and we kind of overlaid them in this graph here, looking at what time the reconstruction weight is active and at what strength, and then also calculating the average spike over time as well. And we can see that they very closely match each other. So we're picking up, what that means is this archetype is picking up the same behavior that this ROI is picking up. So there's another kind of validation of the model. However, our archetype treats these clusters all as one network, where the ROI treats it as separate cells, separate regions. Okay. So I want to, quickly talk about some other experiments that we did in the lab using nanoparticles. And so nanoparticle experiments are important for research in neuroscience because there's a lot of different things that we can investigate. One, for example, is like signaling behavior if the nanoparticles are kind of absorbed into the neuron. And then also like signaling behavior if the nanoparticle adheres to the cell membrane. And so we can look at things like dampened electrical activity. All of those sorts of things are very important. And so we applied some nanoparticles to some cell cultures, different sorts of nanoparticles. So the nanoparticles that we used, they all have an iron oxide core with different sorts of coatings or charges, depending on the type of nanoparticle. And what we were hoping to be able to do from these experiments is to determine whether or not adding these nanoparticles is going to affect calcium signal. And if we can pick that up using the archetypal analysis method versus the spike train method. So our results, I guess I should say, to perform these experiments, first we imaged the Petri dishes without the nanoparticles, then we applied the nanoparticles and then imaged them later in time. And so we can see some results here, some different sorts of behaviors by looking at the mixture evolution index on both sides. So without nanoparticles and then with nanoparticles. Sometimes the behavior isn't quite apparent if there's a change or not, but other times we can pick out some really interesting sorts of behavior. So for example, down here, we have this cell cluster kind of in the center of the frame here in this archetype 9 frame. We can see down here that that frame is active, or that archetype is active at certain times in the video, and that kind of corresponds also to the firing behavior that we would expect to see in our mixture evolution index. And comparing this frame to the other frames here, we see that we have kind of like a center cluster and then an outside kind of spread here. So we expect to see some firing behavior, or we expect that we're seeing firing behavior for that center cluster here. Whereas after we've added the nanoparticles, we don't see that sort of firing behavior. Okay, so we're almost done on time. So just to kind of sum everything up, the challenges for this project were that we needed to have a good biological interpretation of our computational elements of our machine learning model, because we had important biological questions that we wanted to assess. The videos were too large to keep in memory. And also, the machine learning algorithm was slow for large data sets. So those were all kind of challenges that we faced. In my dissertation, I created a method that applies data sketching by projecting the data points into a lower dimension. And then we built some theory around that projection and that method. And then what we didn't talk about today is applying a primal dual hybrid gradient algorithm to the constrained optimization problem in archetypal analysis. And we'll talk about that briefly during my defense. Thank you everyone for coming and listening to my talk. I'd like to also thank my advisor, Dominique Zozo, everyone at the Kunze Neuroengineering Lab, Anya, Derek, Connor, Zeynep, of course, everyone involved in the Kopriva Foundation and the Montana Peaks Foundation, Lisa Davis, John Barkowski. Here are the references that I have. Thank you all very much for listening.
Thank you.
Yay.
Nice talk, Catherine. So, okay, now we are open for questions. Please unmute yourself and ask questions.
I have a question. Catherine,
Hey, Mark.
you there? Hi.
I'm here.
So you've got 7,000 frames of data temporally, right?
Yes.
Roughly, roughly how many calcium spikes can you get into that?
Roughly how many calcium spikes? So, well, okay, so we have 7,000 frames of data. I believe, if I remember correctly, we were imaging at 3 frames per second, and each calcium display, right, each calcium kind of luminescence for a firing event would be, we would expect about, I think, was about 3 seconds of expression. So
quite a few then.
Quite a few.
Yeah.
Quite a few.
So my follow-up to that would be, was there some reason for this temporal resolution? You know, like maybe make the frames happen much more frequently, you know, and get a higher resolution of the actual calcium spikes, et cetera.
Yeah. So we for this project, we didn't apply, we tried to not change the data set too much, but I think certainly you could take the video, do perhaps some averaging or something over short time periods, and still kind of like pick up some temporal behavior, or just look at a short section of the video, not all 7,000 frames, for example. So if you're watching the video and you're like, oh, okay, around minute 3, we have a ton of firing behavior. Maybe we kind of cut the video short around minute 3, for example, and so we have less frames to analyze. We might be able to pick out some even more interesting archetypes. And then the temporal behavior, actually in the reconstruction weights, I think I forgot to say that, but like, yeah, so the archetypal analysis splits the data into spatial component and temporal component. Archetypes are the spatial component. and then the reconstruction weights are the temporal component. So you might be able to get a little bit more information that way, because basically what you would be doing there is you would be shrinking your data cloud. So using the same number of archetypes, you might be able to pull out a little bit more from there, if that makes sense.
I had a question. You had mentioned something that you referred to as a background archetype.
Yeah.
And I would imagine that in addition to, and I assume by that it's just the background of taking the data. But could there also be a housekeeping archetype where the cells are refreshing themselves and it has nothing to do with information transfer?
Absolutely. Yes, that's certainly possible. So what I didn't mention was we also did, you can also apply certain sorts of pre-processing to your video frames to perhaps like eliminate some of those sorts of things. So maybe you have some like image noise, or maybe, for example, one of the things that's really common in processing these sorts of videos is to remove kind of the background luminescence. So you kind of like take an average frame and you just subtract that average frame from each video frame, because really what we want to capture is like a big change, not necessarily the average, if that makes sense.
Yep.
So what I meant by background is basically just like what kind of like for this application would be like what happens at rest. Does that make sense?
Yeah.
More questions? Any questions for
Hey, Catherine.
Catherine?
I
Hey,
had a
Jesse.
quick question.
Yeah.
Hey, Catherine. Oh yeah, you probably mentioned this and I missed it. But how does archetypal analysis differ from non-negative matrix factorization?
Great question. I did not mention that. They are very similar. Go back to the slide. Here we go. Okay. So non-negative matrix factorization would treat this as one matrix here. So you would have basically, yeah. So non-negative matrix factorization looks a lot like this model, but it's a constrained version of this model where the Z matrix and the A matrix have to have positive values. So it's kind of similar. It's almost like I think of it as a more constrained version of non-negative matrix factorization. Yeah.
Right. It's a more non-negative version or something strange. Yeah.
Yeah.
You have two non-negative, these two non-negative matrices going into it. Okay, so
Right.
it is a subset of non-negative matrix factorization. Cool.
Yes. Yes.
And I did have one more, not a follow-up question, a tangential question.
Yeah.
So you have large data, but actually, if you could, and you mentioned it's like too big to fit on a machine, but really 120 gigabytes is not really that big. You can get terabyte machines and stuff. And how would that change this approach if you dumped it all into one machine?
What I specifically meant, was it's a little bit too large for actually MATLAB to process. Not necessarily like you can hold it all in core, but like to actually do the analysis, because we have some pretty big like matrix multiplication happening. MATLAB does not want to behave properly. So the main kind of like the main difference is time. So the standard kind of complexity to solving this problem is on the order of the size of your data, the number of data points, multiplied by the number of archetypes that you choose. When we reduce the size of our data points dramatically using the projections, that complexity reduces significantly. And so there's kind of also another power, just applying the sketching makes the algorithm go faster.
Okay, I gotcha. Cool.
Yeah.
Thanks.
Are there any more questions?
I had a quick question regarding possible like applications of your algorithm. So
Yeah.
based on the archetypal analysis, you're able to kind of drive like main components that make up the network activity. Are you able to also like take that data and then potentially generate a prediction based on like a certain stimuli? So like in your later slides, you had like a stimuli under magnetic fields and then no magnetic fields. So could you take that data and then from your archetypes, could you kind of build a model saying, okay, under this specific stimuli, we would expect these networks to reoccur?
I think as long as you had a way to, okay, so I guess if I'm interpreting your question correctly, you're saying if we grow the cells in a certain pattern, would we be able to say that this model's pattern would hold for growing another set of cells in that same kind of physical formation? Is that?
Maybe not as that complicated. I was thinking more on like a temporal time scale. So like if you had a petri dish with like the same cells and you're looking at like a network activity and then you introduce a new stimuli, could you kind of predict like how that subset of cells would behave under that stimuli?
I think there's definitely a way to do that. I'm not sure if just a direct application would work, because I think it would depend a lot on the stimuli that you're introducing, because there's going to be some biological significance based off of the stimuli. So I think it would vary in that sense. But certainly what you could do if you have some like long time, like a long time series of the imaging, you could, what might help is you could kind of take subsections of video and apply the model to subsections of video and maybe get like a representative pattern of like temporal firing, and then maybe assess it that way. So I think there's potential there, Hamad. I'm just not totally clear on exactly how you would do it, but that's a really interesting question.
Yes. Okay. Thank you.
More questions? Well, if not, thank you, Catherine. Thank you very much for a great talk, and we'll see you all, I guess, in the fall. Thank you.
Thanks, Catherine. Great.